Okay, this strand here would be a single strand of DNA. When we do a sequencing reaction, we're actually adding double-stranded DNA, but then we heat up the DNA and it splits in two. And when we do a sequencing reaction, we are either reading this one or the other one. It is which one you read depends on which sequence do you have. Do you have some sequence of this strand or of the complementary strand? So in this case, I can tell you that the PCR primer that I gave you, which is this guy here. So this is a short sequence of DNA. And the way this happens inside of the reaction tube is the double-stranded DNA gets heated up, separates. And these are in the reaction too. And then we cool it down. So when you cool down DNA, redoubles up, rejoins. But if you have the PCR primer, then the PCR primer will, by Brownian motion, look around where does this base complement this base. So one base complementing the other base, they make a hydrogen bond. But if the other one doesn't complement, then they come apart. So they basically are like dating, right? They're like, you know. And then when they all complement each other, like, oh, you complement me. And then that's when they, let's say that they get married. But what they're doing is they're forming hydrogen bonds, and then they're going to make more of themselves. So, all right, the job you have is to figure out where does this line this up? Is it here, 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 here? And also you have to see, is it on this side? Is it on this side? Is it like this? Is it like this? So the cues for you are, okay, this is 5 prime. This is my 3 prime end. And I made it bigger in here. The hydroxy, that's where that's the one that's going to react with this guy, and then it's going to grow. So first you need to, well, they're going to react with the new incoming nucleotide, not with this. So the first job is to think, okay, if this is the hydroxy Dr. T marked me, and we know that DNA grows in the 3 prime end, then I know this is my 3 prime end, that's my 5 prime end. So if I try to do like this, then I'll have a 5 prime to a 5 prime, and a 3 prime to a 3 prime. Then you know it's wrong. Then like, okay, like this. And now you have to see. So it's written in there. Okay, blue is G, yellow is T, green is C. So the G complements a C. So the blue complements green. So now you have to see where do they complement. And once you find where, you leave it there. I don't know, I'm going to make it like this. It's wrong, but I want you to do it. That guy would make a double bond or make a bond here because G and C complement each other. And then this and this, they don't. So, okay, they would break up. But let's say that they don't care, that they are not compatible, then stay together. Now, so I'm doing this wrong. You're supposed to find where they align. And then you would go like, oh, I'm supposed to grow that way, right? So to this side, because the new nucleotide gets added here. Which nucleotide should I add? I don't know. I don't have the information. So that's another cue for you that this is the wrong alignment. But what you need to do is, what if it aligned here? Then you go, let's say this was right. It's not. But a G and a T, or wait, red is A. A G and an A, they don't actually make hydrogen bonds. But if they did, this is how this would line up. My next nucleotide would come on this side. And so green is C. I would need to find a G, which is green. So then I look on my cards of nucleotide, and I'll find, okay, I found a nucleotide that is green, and I would put that there. It doesn't matter if it's the dideoxy or the deoxy, and you can tell the difference by, okay, these are the same. let's see, where is the dideoxy? I made it darker, and I also wrote the, here, let's see. See, this one and this one are very similar, except that this one has an H here, this one has an OH here, and then I made the dideoxy marker just to try to say, this is the one that would be labeled with a fluorescent dye, and that's the one we're going to read. So your first step on this game is to find where the primer aligns with the DNA. The second step is to see which nucleotides get added here. You add at random. Then there will be 4 new strands being made because I gave you 4 primer molecules. In reality, you make, there's like 1 tera number of molecules of primer in a sequencing reaction. So, but I won't give you 1 tera, I'll give you just 4. So you're going to make these growing strands, and they're going to be only 1 nucleotide added, 2, 3, or 4. So do that. I'll check on you either in 5 minutes, or if you speak up, you write on the chat saying, I finished. Okay, so make the new strands, and then I'll ask you what's the sequence, and you're supposed to read from smaller to larger. So they're going to be, if you added the deoxynucleotide here, then this will be a shorter one. If it was a deoxy, another deoxy, and then a dideoxy, that's the longer one. All right, give it a try.